Hi, I'm Heather Dame. I am a graduate student at Central Michigan University, and I have been helping out with my advisor, Anna Monfils, on her project with aquatic invasive species.
Aquatic invasive plant species are a real big problem in the Great Lakes, and there's a concerted effort across the region to work with other states to see what is coming into the state, to have an early detection of those and a rapid response. And what our project is going to be able to do is give a better idea of how to respond to invaders as they're coming in, look to possible eradication, and help to keep our Great Lakes waters as close to their native diversity as possible. The goal of our project looking at aquatic weed management is really to figure out what's working and what's not working, and then see if we can ramp what's working up to a scale that can really combat the presence of some of these invasive species. And Heather's contribution is really going to be an understanding of what is going on below the water, what species are there, which is something that had not been archived previously, and then looking at the relative impact of different management strategies, which is going to inform sort of management strategies down the road.
So there are several different methods, such as herbicide treatments and harvesting of aquatic plants and placing benthic mats. So what we want to do is look at these different methods and try to figure out which ones work, to what extent, and which ones work better than the others and why. The way that we go about answering these types of questions is by actually getting into the field. So what that involves is us going out before the treatments occur, looking at the plants, making general observations, taking some photos. We have underwater cameras, of course, and we also take biomass sampling. So we measure out a 10 by 10 meter area and look specifically within this area. We do transects and collect biomass samples. We can capture some of the plant diversity and the percentage of plants that occur in that quadrat and figure out what are there before. So then after treatment, we can go there and do the same exact thing. Both times we're taking water samples, soil samples, temperature, pH, conductivity, all these readings, so we know exactly what's happening before and after. We're also taking general observations of just measuring plant growth of both the target species and other plants in the area. While we are in the field, we collect specimens to help us identify the plant species that we are seeing while we are underwater, which is very valuable to us because when we write in our notes a certain species, we can go back to the herbarium and know exactly the type of plant that we're looking at. When we get back to the herbarium, then we press the plants and dry them and deposit them into the herbarium as reference specimens. We can then identify them using several different field manuals and online resources so we can get an exact identification of that specimen.
The data that Heather's collected for her project, she's collecting voucher specimens that will be deposited in the Central Michigan University Herbarium. CMU Herbarium is one of the participants in the Invasive Aquatics project. The specimen will be digitized and data will be transcribed from them, and they will be entered into two different portals. One's the Midwest Herbaria portal that's been established for general collections without the Midwest, and then specifically it will be going into the website that's devoted to the Invasives project itself, where we'll be collecting specimen information from the different participants in the project, and it'll all be available in a common site, both animals and plants. So her specimens from her sites will be a part of the bigger picture and available to anybody doing research in that area, so that it will be one little part of a bigger puzzle.